Welcome to the model organism session. I'm Riku. I'm a first-year grad student here. So model organisms are organisms that are commonly used in research. And here's like some common examples: bacteria, bacteriophage, E. coli, sea urchins, fish, mice, plants, all sorts of different kinds of organisms. And these are commonly were chosen for practical reasons. So they're easy to maintain, easy to keep in the lab. They're cheap, they're robust. They have short generation times. They give many progeny, so you can do tons and tons of experiments and screen through tons and tons of animals. But what I think is most remarkable about modern organisms is the kinds of genetic tools that are available in them. And people through the decades have worked on tons and tons of these very clever and sophisticated tools. And these tools allow us to do very precise experiments and answer very specific questions. So today we have three speakers who are going to talk about their work in model systems. But before that, I figured maybe I'd just introduce one type of tool that we can use. So right now I work in zebrafish, but previously I worked in Drosophila, which are fruit flies. And in Drosophila, there's tons of just amazing genetic tools. One of them being this Gal4 UAS system. So Gal4 UAS was first discovered in yeast. So it's a mechanism by which yeast activate genes in their genomes. So yeast make this Gal4 protein, and this Gal4 protein binds specific sequences, which are called the UAS, and basically drives transcription of whatever gene is downstream of this sequence. So in 1993, Andrea Brand and Norbert Perimon, two very clever scientists, they decided to co-opt this yeast transcriptional system into Drosophila. So what they did is they made a fly where you can express Gal4 and pretty much whatever cell type you want to. And then you cross that to another fly that has a UAS and then any gene you want to express. So this could be your favorite transcription factor, cell adhesion molecule, RNAi, anything. So the flies you analyze basically activate this gene and whatever cell type you want. So tools like these really enable sophisticated analyses, genetic analyses, and there's tons of really cool tools out there that our speakers are going to talk about today.